Sharat Varma is our next speaker. He is an engineer, too, in cell culture process development at Genentech. His experience includes late-stage mammalian cell culture process development for both antibody and non-antibody molecules, mainly focused on studying the effects of media components on product quality of recombinant proteins. He has a bachelor's degree in industrial biotech from Anna University in Chennai, India, a master's in biotech from the University of Pennsylvania. And the title of his presentation is How to Establish Cell Culture Parameters for Improved Product Quality.
And today I shall be presenting some of our findings on some of the cell culture parameters and how they affect product quality of the antibodies that we produce. So first, I'd like to kick off by acknowledging people who have contributed to this work. The key contributors are first Natarajan Vijayashankaran, my manager. Yi Yang is our analytical colleague who has contributed significantly not only to analysis of samples, but also to development of hypotheses testing. Rick Chen was an intern who has also significantly contributed to this work, and Steve Maya, our group manager, for all his leadership and support. This work has been extremely collaborative, as it involved a lot of my heads being put together, so I'd like to thank a lot of people in cell culture, besides which I'd also like to thank our purification and analytical counterparts for processing a ton of samples, and ditto for our pilot plant and media prep and analytical folks for doing a ton of work. So going on, I will first briefly talk about the cell culture process here that we usually use at Genentech, and then go on to talk about bulk color. That is the product quality attribute that I shall be talking about today. Why is it important? How do we control it? Probable mechanisms by which bulk color is caused, and some novel methods to reduce bulk color. And of course, go ahead to summarize it all. At Genentech, we typically use a fed batch cell culture process. For people who are not familiar with it, we start the culture with a basal media, and then after a few days of growth, add extra nutrients to further keep the culture going on. Of course, the product that we're interested here is the final therapeutic protein that we produce, which typically is antibodies. In this particular case study, we have an older process which used peptone-containing media. As many of you may know, using peptones has a lot of issues with it, including lot-to-lot variability of the peptone, as well as a high risk of contamination from advantageous agents. In the newer process, we wanted to use a chemically defined media so that we can avoid these risks. And at Genentech, the platform process, the concentration of the media is kind of optimized based on growth and productivity, because obviously everything else will change according to cell line and the process. And typically, our platform process yields between 3 to 5 grams per liter of the protein of interest. However, in this particular case, since we have an older process, establishing product comparability was very important. And in establishing product quality, I wanted to specifically talk about bulk color. So this is the color of the final drug substance that goes into the patient. And typically, color has not been a big issue over the many years, but off late, with higher concentration formulations going on, becoming the trend in the industry, we're seeing that this has also become an important product quality that we are concerned about. For example, you can see in this picture, the drug substance, the final product that's actually going to the patient, differs significantly between the old process and the new process. Now, typically, the FDA uses the COC assay, which is the clarity, opalescence, and coloration method, which is derived from the European Pharmacopoeia, to assess what the color of the substance is. Again, kind of briefly talking about what the COC assay is. Simply put, it's a comparison of your drug substance at whatever concentration you intend to use it in the patient compared to a standard reference. There are standard references in different color series. This is brown for B, BY being brown yellow, and Y for yellow. There's also green yellow and red series, which I'm not showing here. And each of these color series has serially diluted samples. So you basically take your sample and compare it visually. So it's done by a technician who's trained in this and visually compares it against these samples and says, this is less than or equal to a certain other standard on this. However, this is always done on the bulk formulation, and especially for higher concentration formulations, it is impractical to use it for small scale studies, which is where we do most of our process development, either because we don't generate enough material, or that we need a lot of resources to process all our small scale material all the way down to the final formulated bulk. So we first had to develop a surrogate assay to test for color. The other disadvantage with using this COC assay is, as you might say, this color of my shirt is blue, but who knows if it's navy blue or royal blue. And that's always a case of these samples also. There's a lot of subjectivity in the rating of the samples, even by the COC method, and it's not quantitative enough to detect minor variations that may occur between two reference samples in this. So we also wanted our surrogate assays to be quantitative. So how else do we measure color? We have two methods that we've developed which are quantitative. The first one here is the normalized intrinsic fluorescence tool for yellow-brown proteins, long name, short for NIFTY, and it's developed by Yi Yang and Suguru. Basically, it looks at the absorbance at 280, which is a measure of concentration of protein for the sample versus a reference sample, and then looks at the fluorescence at a certain excitation emission. And as you can see in this image, for the same concentration of sample versus reference, you can have very different fluorescence values. So by normalizing this fluorescence to the concentration for both the sample and your reference, we get a relative concentration normalized fluorescence, which is what we call as NIFTY. The other tool that we use is called total color, which is developed by Trevor Schwartz. It tries to mimic how the human eye perceives color. And we first take a UV visible spectroscopy of the sample and then convert it onto this LAB scale. Now, the LAB scale is used by the industries that deal with color, for example, the paint industry, to assess what their shades of color are. So we're just adapting it and using it for proteins in our case. The LAB scale consists of a three-dimensional space, which I've represented here as L on the third axis here. L stands for luminosity, and it basically gives you an idea of how intense the color is. Whereas the A and B form a plane of all the colors that the human eye perceives. And in this particular plot, we've plotted out the COC standards that we showed in the previous plot as red, brown, brown yellow, yellow, and green yellow. And in this method, we're able to pinpoint where the sample falls on this plane. So this method actually not only tells you how intense the color is, but also what shade of color the sample is. And that way actually mimics the COC assay itself much better. And so we also have a readout of a predicted COC value. Now, okay, we have a method of measuring color, but then we still have an issue of going back all the way to the bulk. So we wanted to simplify that, reduce the resource input into it, and we want to use the Pro-A pools, which is the first step of purification for our cell culture. Typically, cell culture fluid goes to Pro-A and then goes on to all the other steps of purification before it's finally formulated. Like I've already mentioned, we use the COC method, the standard compendial method, for looking at color at formulated bulk. However, we're able to use the NIFTY method, which I just described in the previous slide, both at the Pro-A pool as well as the formulated bulk, because it is a concentration normalized method. It's independent of concentration. What we also do, find out that we can use the total color method at the Pro-A pool, except that we have to concentrate it up to the same concentration as the formulated bulk itself. So this way, we're able to use both the NIFTY method and the total color method on the Pro-A pool, thereby reducing a lot of resource that we put into assessing color. Okay, now we have a different method of assessing color. We have a different pool of assessing color. So how do you know we're actually looking at the color in the formulated bulk, or what we look at in the small-scale experiment is actually going to translate finally into the large-scale work? So first, I just want to show you what the correlations we found are. On the left here, I have the total color used on both the concentrated Pro-A pools as well as on the final bulk in whatever cases we have. And you see a very good correlation between the two. So this clearly tells us that the Pro-A pool color is indicative of the final bulk that we generate. Yes, we will also notice that the total color is always higher in the Pro-A pool compared to the UFDF, but that is to be expected because the Pro-A pool is not as clean. On the right side, I have comparison of the three assays, the NIFTY, the total color, and the COC being marked by the different colored markers on this plot. And once again, we see a very good correlation between the three assays in this. This again gives us very good confidence that we can use either NIFTY or total color as indicative of what is going on in the final process. So going on, I first want to orient you all to what I'm going to show in the subsequent few slides. All these slides, I'll be discussing what cell culture parameters, specifically media components, affect color. And all of them, in the top left, I will have final volumetric IVPCV, which is the indication of growth. The final titer on the top right, which is indication of the process productivity. And on the bottom left, I'll have a color, measure of color, either total color, as in this case, or NIFTY. On the x-axis of all these plots, I'll have the factor that I'm discussing in that particular slide. However, I do want to point out that all our experiments are multifactorial, full factorial experiments, but for sake of simplicity, I want to discuss only one factor at a time. All other factors that were tested will be plotted out in legends. So going to the data on this slide, we found that the B vitamins in our media can significantly contribute to color. If you look down here, you can see that by going from 100% or the standard media that we use all the way down to 0%, we actually reduce color very significantly. We almost halve the color that we get in our process. Of course, we do see a concurrent reduction in growth as well as titer, but that is to be expected. The vitamins are there for a reason. The next factor that we looked at was iron. And here on the x-axis, I have two factors actually. One is the type of iron that we tested out in cell culture, ferric citrate, ferric nitrate, and ferrous sulfate, and then the concentration at which it was tested, low, medium, and high. Looking at the color here and just focusing on the ferrous sulfate cases, you can clearly see that with a reduction in the ferric sulfate concentration, we see a reduction in color. Again, you can see a reduction in growth as well as titer, just as we saw with the B vitamins. But what is very important here is that the ferric citrate had lower color than the ferrous sulfate at equimolar concentrations. And this came with an advantage of actually having better productivity than the ferrous sulfate cases or ferric nitrate cases. Next, we looked at substituting cysteine with cysteine. The monomer form was substituted with the dimer form in our media. And once again, we see a reduction in color. Now, if you notice the plots, this is probably not a very large reduction in color, but we definitely believe that small drops make a mighty ocean. This, of course, came with an advantage that we actually got higher titer, so that's actually a good thing for us. The next factor that we tested was the addition of hydrocortisone in our media. And we see that with the addition of hydrocortisone, we do get less color here. And the titer improves based on addition of hydrocortisone. Now, through all the work that we focused on looking at reducing color, what we also saw was that acidic variance was going down. Acidic variance, as many of you may know, is a pretty important challenge that many projects face. We always try to keep it pretty low. And what we saw was that not, as a double bonus, by reducing color, we also reduced acidic variance. Now, all this seemed to not make a lot of sense because we didn't even know what the mechanism of causing color was. But over the many studies that we have done, and with Yi-Yang's hypothesis that we have developed, this sort of thought process. And it's a very busy slide, but I'll try to simplify it. We believe that the main mechanism of color formation is the oxidation of amino acids on the antibody or therapeutic protein that we are producing. And now it kind of all falls into place where all the components that I just talked about, be the vitamins, the cysteine, cysteine, iron, or hydrocortisone, all in green here, all have some role to play in the oxidative stress environment in the media. The oxidative stress environment here, reactive oxygen species, are denoted in the red here. So they all kind of come together by using this hypothesis. Of course, there are other mechanisms by which, especially B vitamins, can directly interact with the protein, covalently or not. However, we definitely believe that oxidation is the bigger player here because B vitamins and iron, both of them, have the biggest effects on reducing color. Now, of course, the next question is, we're always losing titer when we try to reduce color because we're taking out the vitamins, we're reducing iron and things like that. So how do we keep the productivity high? And since we believe that oxidation is the method of that color is produced for the most part, we should use antioxidants. It seems like a no-brainer. However, doing an antioxidant screen on any media is not a simple thing, especially in cell culture. Variation by cells, variation by media, variation by process conditions, things like that. So we needed a simpler system by which we could actually screen a lot of antioxidants. And what we realized from this, just looking at such a plot, was that most of these reactions are just spontaneous and they're cell independent. So why not just do an in vitro antibody media incubation study? What we basically do is take media of interest with whatever components we want, spike in a purified antibody and incubate it over a period of time. And we said, okay, maybe that will actually tell us what's happening in the cell culture process itself. The advantage would be that it's a very easy setup. We can always do a lot more cases. And we applied this model to hypothesis development and testing. And here I'll just show one example of such an experiment. Going back to the oxidation hypothesis, we know that vitamin B can be photooxidized to peroxide and superoxide, and iron can react with the peroxide in the Fenton reaction to give us hydroxyl ions. We also know, of course, that the catalase can cleave away the peroxide to harmless products. So we did a full factorial experiment in the media incubation model looking at these three factors. And here I show you the data from that. we see that without the catalase, with an increase in iron and vitamin B2, we see an increase in color. This mirrors exactly what we saw in a cell culture experiment, thereby giving us better confidence that the model actually works. It is representative of what is happening in cell culture itself. However, how it goes one step further is by giving insight into actual mechanism. With the catalase, the effect of iron on color is basically nullified. You can compare between these two plots here, whereas the vitamin B2 still has an effect. So it does go back and tell us that yes, iron may be reacting or causing color mainly by the Fenton reaction. Okay. The next one was we went to do antioxidant screening. We looked at literature for about 50 components or so, used the incubation model, and then screened for compounds, and then went to cell culture experiments using those compounds. Quickly looking at the in vitro media experiments, 0 basically means it's the control, and anything in the negative means it's a lower color. We found multiple components that work, but specific interest was carboxymethylcysteine and hypertaurine because they gave us the same reduction in color as all the other media modifications that we had made to the process. So we went ahead, took it to cell culture. I'm sorry this is not rendering very well, but I hope you can see that the components did not affect cell growth here in PCV or the productivity here titer, but did reduce the color significantly. And hopefully you can see that the hypertaurine and carboxymethylcysteine cases actually ended up lower color than the controls. And with that, I'd like to summarize that product color is a very important product quality attribute which is getting attention only recently because of the shift in the industry to higher concentration formulations. We identified multiple levers that can reduce color. We also see that acidic variance lowers with the lower color. There are multiple mechanisms for color formation, of which we suspect that oxidation is the biggest contributor. And of course, we identified many antioxidants that can reduce color without compromising productivity. And with that, I would like to thank you, and I'll take any questions and comments you have. Yes?
Did you do a test on, since this is chemically defined media, did you do any test of adding hydrolysate back to see whether it would revert the color back to the original form?
Yes, we have done tests on hydrolysates. We do see a reduction in color, but that is extremely specific to this particular cell line model. We have seen that different hydrolysates have completely different effects on the same molecule, and they are not molecule specific. Like the same peptone, if you see in model X, reduces color. In model Y, it seems to do nothing or increase color. So it's extremely dependent on how the hydrolysate contributes to the metabolism of each cell line being tested and the product that's formed.
Yep, I think we have time for one more.
Do you specifically know which amino acids it's oxidizing? And if you know those particular amino acids, did you do a test of adding those specific amino acids to have like a surrogate amino acids that would get oxidized, and you can use that to protect your protein of interest?
So we don't know for sure of our the amino acids that are getting oxidized in our protein. The hypothesis has been developed from stressed samples. They're not exactly the same from the cell culture. But then on the other, to answer your second question, our media already has all those amino acids. The media has all the 20 amino acids that cell culture uses. So, and they're pretty much at a high level also. So we don't see why just having a surrogate amino acid in the media will help. Okay, thank you very much.